Hello there, welcome once again to our science lessons. So just to choke our minds, you remember that in our previous lessons we talked about microorganisms, and we said that microorganisms are very tiny organisms, and for that purpose they cannot be seen by our naked eyes. And for this reason, we must find aiding equipments or apparatus for studies. And one of them is very common in a school situation, which is the light microscope. But in advanced areas or in advanced studies and research, we usually use an electron microscope. Microorganisms were of importance in that lesson. We said there are different categories of microorganisms, and if you can just remember, we said we have bacteria, we have fungi, we have the protozoans, and then we have the viruses. And we were able to find out of these four groups, nearly all of them cause diseases. But majorly the diseases that affect human beings are due to bacteria. And then we went ahead to find out that those microorganisms that will cause diseases in our bodies, we were able to classify them as pathogens. So any disease causing microbes that will invade our bodies and then cause malfunctioning of different body parts and systems, we refer to them as pathogens. But again, we said for these microorganisms to cause diseases or to be classified as pathogens, they must be transferred from the source to the body of an individual. So if we have microbes and they are classified as pathogens, they cannot just cause disease if they are independent of the body. So they have to be transferred from the source which is producing the pathogens to the body. So we have different parts of the body where the pathogen will encounter the body. It could be the outer part of the body, which is the dermis or the skin. Now, exposure of the body just to pathogens on the outer parts, most of the time it does not cause diseases unless it gets into the inner parts of the body. Inner part of the body means, for instance, the circulatory system. So that process whereby pathogens are moving from the source into or to the body, we call that process as transmission. We were able to find out that process of transferring pathogens from the source to the body is called transmission. And again, you can remember that we had two types of transmission. We have direct transmission. And in this case, direct transmission means if, for instance, you touch somebody or you get into contact with somebody who is suffering from a disease, you are able to get that disease. Very common is Ebola, a very contagious disease. So those diseases that are transmitted directly through contact, we call them as contagious diseases. And then two, we were able to find out that sometimes these pathogens or microorganisms could be transmitted through other medium. or through other media, for instance, through air, through water, through food, or through other particles. For instance, through air, when somebody sneezes or coughs, is able to release droplets in form of saliva, in form of maybe mucus, which is then transmitted or which will carry the pathogen to the other person. So we were calling this as indirect transmission. Now, slightly after the pathogen has encountered the outer part of the body, sometimes it will get into the body, or the skin will be enough to prevent the entry of pathogens into the inner parts of the body. But if it succeeds, if that pathogen succeeds to get into the inner part of the body, we call that process as inversion. Inversion. So we shall say the body has been invaded by a disease. Now, after inversion of the pathogens into the inner parts of the body, we usually expect two reactions. One of the most quickest reaction is what we call an inflammatory response. Inflammatory response. Inflammatory response. So again, in our previous lesson, we talked about inflammatory response. We said inflammatory response is that quick response or response of the body towards or to pathogens or any harmful stimuli. Pathogens is an example of a harmful stimuli. Inflammatory response. Response of the body system to presence of harmful stimuli. So here we need, the most important factor is presence of harmful stimuli. Remember, we talked about stimuli. We said stimuli is that change in the environment, and the change in environment is the presence of that harmful stimuli inclusive of the pathogen. So if you can just remember the role of inflammatory response, we said the role of inflammatory response is to remove the source of injury. Sometimes when pathogens get into our body, they cause injuries to our body. So to remove that source of injury, we shall have the first response, inflammatory response, and it usually involves immune cells. It is going to involve immune cells with other parts like blood vessels, and without forgetting that we mentioned mediators. So we shall not go into details of this because we have already known. We are just reminding ourselves that inflammatory response is that response of the body toward harmful stimuli, which could be an example like pathogens or others damaged cells or necrotic cells. So the other response that comes in handy to tackle invasion of the body by pathogen apart from inflammatory response is the immune response. Immune response. Immune response. Now, up to that, then it gives us a platform for today's topic. Our topic today is immune response. So I wanted us to flow from the microbes up to the immune response so that we can now begin from there. So now you can appreciate how microorganisms are bringing about these two processes, and we have already looked at one, which is inflammatory response, then the second one is immune response. So today we look at immune response. So today, as we look at immune response, we are just classifying it as one of those responses that occur in our bodies once the body is exposed to a pathogen. So we have said one of the first response is inflammatory response, but inflammatory response is not enough. Sometimes it's not enough to clear the pathogen. So the second process that comes in now to fight the pathogen or to clear the pathogen is immune response. So in this case, we have to find the meaning of immune response. Immune response is that process by which the body responds to presence of a pathogen. Is the process by which the body responds to presence of a pathogen. Presence of a pathogen. And the reason for respond is to actually destroy the pathogen, either to deactivate it or to destroy it. In the process, destroying the pathogens. But it has to be noted that immune response does not just act on pathogens, but any other harmful stimulus that will be exposed to the body. So in a nutshell, we call those harmful stimulus that will trigger immune response as antigens. So if we factor in antigens in our definition, then we slightly change our definition, and then we say immune response is the activation of immune system when the body is exposed to antigens. When the body is exposed to antigens. So immune response will now kick into activity after the body has been exposed to antigens. So what are some of these examples of the antigens? Examples of antigens include antigens. Before you go to the example, you can slightly define. Antigens are foreign materials to the body. Foreign materials to the body. So because they are foreign to the body does not readily accept those materials. We are going to find out the examples. Examples include, one, we have talked about pathogens, pathogenic microbes. Pathogenic microbes. Microbes are equivalent to microorganisms. So organisms themselves. But sometimes these pathogens produce products. Those products can also be an example of antigens. So pathogenic products. Pathogenic products, e.g. toxins. We have a very important bacteria that causes tetanus. It produces toxins that usually paralyzes the whole body. Microbes could be several, as we have already mentioned, could be a bacteria, but remember, not all bacteria cause disease. Therefore, they are non-pathogenic, but we have most of the bacteria are pathogenic. We have fungi and several like protozoans, like the one that cause malaria, Plasmodium malariae, viruses, the one that cause HIV/AIDS, and we have mentioned pathogenic products. But again, we also have materials in the body that body can respond to it as antigens. We call those materials as self-antigens. 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 For instance, you realize that inflammatory response can arise when somebody produces some hormones. For instance, this is very common in lactating mothers or pregnant women. So hormones could be an example of antigens. The body has produced its own material, but now the body recognizes it as foreign material. So we call those ones as self-antigens. Then we have others called allergens. Allergens. Allergens are antigens that usually causes allergic reactions. Allergic reactions. Allergic reactions are synonymous with inflammatory responses. Good example of allergens could be dust. You realize that some people, when they are exposed to dust environment, they start sneezing. Sneezing is an example of an allergic reaction or an inflammatory response. Remember, we have defined inflammatory response as that response or that activity which aims at removing the source of injury. So dust. So for the body to respond to dust, you are going to sneeze, you are going to cough, so that that mucus that has captured or trapped the dust can be expelled together with dust. Pollen grains. You realize that those people who live close to fields or fields with tussling maize, at that point there's a lot of pollen grains in the air. So at that point you realize that very many people will be producing allergic reactions just in response to pollen grains. Some perfume can also act as allergens. When some people are exposed to some perfumes, they are able to produce allergic reactions. So they form part of the allergens. And then the last example are grafts. Now, what are grafts? Grafts are body parts that are functional, which are meant to replace the parts of either your body or somebody else's body so that it can continue to function properly. A very good example could be kidney. If, for instance, you find that, or the doctors find that your kidneys are malfunctioned, they can take a kidney from one person, and then you are given that kidney. We shall call that one as a graft. But sometimes those parts that have been donated are rejected. So when they are rejected, they form part of antigens. So grafts, not all grafts are antigens, but the ones that are rejected. So graft rejection. Graft rejection. So sometimes maybe you have been injured somewhere, maybe a tissue, only tissue has been removed. That tissue can be replaced from either from another person or from the same body. So when we have graft rejection, we classify it as antigens. So when the body is exposed to these antigens, as we have mentioned, immune system will be activated or will be triggered into activity. So the triggering of immune system into activity, we call that one as immune response. But now we have to find out what type of structures or cells in the body are actually involved in the immune response. So cells involved in immune response include we have what we call T cells, we have B cells, and these two could be either for lymphocytes or macrophages of lymphocytes and macrophages. So these are the main cells that are involved in the immune response. But without actually complicating the names and the rest, these are simply white blood cells. White blood cells. So it usually involves the white blood cells. But the other question that comes into place, then how are they involved in the immune response? How do they function? How they function? Now, they function in maybe 3 or 1 or 2 ways. So most of them, in fact all of them, will produce some chemicals called lymphokines. They produce lymphokines. These lymphokines usually influences the activity. It kicks or triggers the activity of the host cells, which influences the activity of the host cell or host cells. Now, what is this host? The host cell is that body into which the pathogen or the antigen has been exposed. So the lymphokines, once antigens have been exposed to the body, lymphokines will be produced, which now kicks or ignites or makes the cells of the host to start functioning. How will they function? Some of them, like the lymphocytes will produce antibodies, proteinous substances, antibodies. The other name of antibodies are immunoglobulins. So they'll produce antibodies or immunoglobulins, which will now react with antigens. So we are going to find out how these reactions actually occurs, or the kind of antibodies that are produced. So this type of reaction is usually called antibody-antigen reaction, leading to antibody-antigen reaction. So this results into antigen antibody reaction. So remember, antibodies have been produced by the lymphocytes. But before this process takes place, the most important part that happens first is that we have the macrophages, the one that we have identified that they are involved. Macrophages are going to engulf the pathogen. Macrophages engulf the pathogens. And upon engulfing the pathogens, it will now recognize the structure of the pathogens in the process. Recognizing the structure. In the process, recognizing the structure of the pathogens. And then after recognizing it, it's not going to stop there. It will use this information now to produce antibodies. Then uses this information of recognition to produce antibodies. So the major player here in immune response are the antibodies, but we have also other factors that have been involved or other structures that have been involved, so that now the last resort is production of the antibodies. So antibodies are the most important structure of the immune system. And because of that, we have to look at types of antibodies and how they function. Types of antibodies. So we have said it's the antibodies that will now react with antigens. We have to find out some of them. So one of them are called lysins. The word lysine usually emanates from the word lysis. And lysis means to break. So in this case, we shall ask ourselves, what do the lysins break? So after macrophages have engulfed, they have recognized the structure of the pathogen, they have now used the information to produce antibodies. The antibody could be the lysine. Now the lysine, once the lysine has been produced, it usually destroys, breaks the cell wall and cell membrane. Breaks or destroys. Destroys cell wall and cell membrane. Cell membrane and the cell wall. Now, when cell membrane is destroyed, remember the function of cell membrane. In year 7, you are taught or you are told the role of cell membrane. One of them is to selectively allow movement of material, selectively allow materials in and out of the cell. But now it has been damaged, it has been broken, it has been destroyed. What will happen is that now, if we have, this is the cell, or that's a bacterial cell, and it has been, parts of the membrane has been broken, it's going to allow influx of materials. And some of the materials that are going to get into these cells include water. And excess water or entry of water into a cell, it usually offsets osmotic balance. osmotic balance. Osmotic balance is going to be offset. So in the process, the pathogen is going to be killed. So it began with the destruction of the cell wall, if it is a bacteria, because bacteria, fungi, and even sometimes viruses, they usually have cell wall and then cell membrane, allowing water offsetting osmotic balance, and in the process, death of the pathogen arises. So this is one antibody. The other antibody is called 2-agglutinine. Agglutinines. Agglutinines comes from the word agglutination or agglutinate. Agglutination means to clamp. So what will happen to the cells upon production of agglutinines is that if we have cells, these are bacterial cells that were apart, presence of agglutinines will cause them to clamp together. They clamp together. Those are cells that have clamped together. If these are bacteria, remember one way in which bacteria reproduces is through binary fission. Fission means split. Binary is 2, splitting into 2. So if these cells have clamped together, how are they going to split? If you try to split, there is some adherence there or some barrier because of the agglutination. So in the process, the clamping together of these cells will interfere with the reproduction. So reproduction will be interfered with. And at the same time, when cells clamp together, it becomes very easy for the phagocytes, remember the role of phagocytes, to engulf. It engulfs. So the phagocytes will easily destroy. They are going to be destroyed very easily by the phagocytes. So in a nutshell, we are saying agglutinines, they cause the pathogens to clamp together. When they clamp together, it's going to interfere with the production, and also it becomes very easy for phagocytes to destroy them. So if we may make a note, we are saying agglutinine causes pathogens to clamp together, and interferes with reproduction, and becomes an easy target of destruction by phagocytes. Remember we talked about phagocytes, those white blood cells that will usually engulf. If that's the pathogen, it will engulf, forming a vacuole, food vacuole. When that food vacuole is formed, they release lytic enzymes. The lytic enzymes will now destroy the pathogen. So we have lysins, agglutinines, and then we have opsonines. Opsonines, they cause destruction of the pathogen by sticking, adhering on the surface of the pathogen. When it adheres on the surface of the pathogen, then it becomes very easy for phagocytes now to destroy it. Adheres, sticks. on the surface of the pathogen. Sticks on the surface of the pathogen, therefore becoming easy to be destroyed by the phagocytes, and of course by the macrophages through production of other antibodies. And then the other type of antibody are called the antitoxins. Antitoxin. Now, the word itself is self-explanatory. Anything anti means it acts against. So we have an antigen in form of toxin, products that are produced by pathogenic microbes. Some of them we said could be toxic. So if we are going to have antibodies that are antitoxin, they are going to act against the toxins produced by pathogens, and in the process they neutralize. So this one acts by neutralizing the toxins, neutralizing toxins produced by pathogens. So if this is going to happen, then actually they'll have acted against the antigen. So we say we usually have what you call antigen antibody reaction through production of several of them. Sometimes it could be one or a multitude of them, the four of them being produced at once, depending on the kind of the pathogen that has invaded the body. So that's how the immune response works. But now, based on that, again, we can classify immune response into two categories of immunity. So we have two major categories. We have what we call innate immune response. And then we have adaptive immune response. So even before we look at each one of them, we can go ahead to classify further. Adaptive immune response can be classified further into two. We have what you call acquired, naturally acquired, immunity, and artificially passively acquired, that is passively acquired. Passively acquired. Passively acquired immunity. Of this passively acquired immunity, we can have active. Active is the end. Active and natural. Active and natural. So we are going to look at each one of them. Innate immunity, adaptive immunity, what it means, and naturally acquired, passively acquired. So let us start with the first one, A. innate. Innate immunity. Innate immunity is that immunity which a person is born with. You realize that people who are born in tropics, especially Asian tropics and African tropics, they rarely suffer from malaria because they have inherited immunity against malaria. But those people from in temperate regions, they are very vulnerable to malaria as compared to those one in the tropics. If, for instance, you realize that if we are going to have a mosquito that is carrying the same plasmodium load, it bites one who is coming from the temperate region and another one from the tropics, especially the Asian tropic and the African tropics, that one of the temperate will develop symptoms of malaria very fast. That one of the tropics will require high load of the plasmodium so that it can now develop the disease or the symptoms of malaria. So in other words, innate immunity is that immunity that is inherited, that is passed on from parents to offsprings. But when you say passed, we must be able to find out how. Passed on from parents to offsprings genetically. Genetically. So in other words, it is inherited. Inherited. So you realize that some people suffer from some kind of diseases, while the others do not suffer because they have an innate immunity. And this is the most important, the most dominant immunity in the body of human beings. It's the most dominant. But despite this, sometimes this immunity, usually as the person continues to grow from childhood to adulthood, the immunity usually wears off. So because of that, it has to be boosted. That's why I realize that even if you have the ability to resist malaria due to innate immunity, sometimes as you continue growing, you become more vulnerable to the same if you are exposed to the plasmodium that cause the disease. So because of that, it does not last for a very long time. So since it does not last for a long time, therefore it requires boosting. It requires boosting of the immunity. How do you boost the immunity? Through processes like vaccination. Vaccination. That's why right now there's a lot of scientific research going toward the development of vaccines of several diseases, despite the fact that we have maybe a huge population of human beings which are resistant to some diseases, but still vaccination, development of vaccine is going on. Like right now, there's a lot of efforts going towards development of malaria vaccine. It is still in course. And then the second category, the two broad ones, we have adaptive immunity. Adaptive immunity. Adaptive immunity is also called acquired immunity. Acquired immunity. And I've said we are going to have different kinds as we shall be able to find out. But what is the main feature the main feature or characteristic of adaptive immunity. The main feature of adaptive immunity is that the body will be exposed. Somebody is born without the disease or without the pathogen, but is exposed to the pathogen after birth, and the body or the macrophages are able to recognize the pathogen after inversion, and after that it is able to produce antibodies against that pathogen. But the structure of that pathogen is stored or retained in part of cells called memory cells, such that when this person is attacked the second time or any other subsequent time, the macrophages or the antibodies will be able to recognize that this is the same pathogen that attacked us one time, and this is what we did. The macrophages will know that this is the kind of antibodies that we produced. So acquired immunity, the body will be exposed to the pathogen. After exposure, it will react by recognizing first the pathogen, storing the recognition information into memory cells, and producing antibodies. So any subsequent attack, the body will just remember, because it's already in the memory, this is the same pathogen that attacked the body, and this is the kind of antibodies that were produced to be able to clear it. So in a nutshell, acquired immunity, the body is exposed initially to a pathogen, and it recognizes the structure whose information, so this information now of recognition information is used to produce antibodies. Antibodies are produced. But it does not stop there. The recognition factor or the recognition information is retained in the memory cells. Recognition signals are retained in the memory cells. Now what for? So that any subsequent attack, the body will just be able to recognize this and produce again the same kind of antibodies that it produced. Any subsequent attack, any subsequent attack, for any subsequent attack, the body produces same antibodies against the pathogens. So this one means that if you suffer from polio, that's what happens with the diseases like polio, measles. We have even whooping cough, the pertussis. What happens? You are exposed to a polio virus or measles virus, and then the body recognizes the virus as the pathogen, will keep it in the memory cells, but at the same time producing antibodies against the measles virus at that time. Then any other subsequent attack, the body will just remember that this is the pathogen that attacked me at one point, and I produced this kind of antibodies. So in fact, at that time, any other subsequent attack, second, fourth, you are not going to produce any symptom of the disease. So you realize that the second subsequent attacks, you are not going to suffer from the symptoms of measles. And then we have different types of this adaptive immunity. We have said we have two types. We have one naturally acquired. So naturally acquired immunity is, in fact, what we have just explained above. Naturally, the body is naturally exposed to the pathogen and it produces antibodies. But the structure of the pathogen is retained in memory cells for use any other time. Body is naturally exposed. Two, is naturally exposed to pathogen and produces antibodies. Produces own antibodies. It could be on, or we just leave it that. Antibodies against the pathogens. So after that, the memory of pathogens, the structure of the pathogen is stored in the memory cells. Is retained in the memory cells. So that now any other subsequent attack, the body will respond in the same way to produce. So the role of memory cells to produce antibodies for any subsequent attack. For any other subsequent attack. So this is the case of suffering from diseases, e.g. suffering from measles, poliomyelitis, a whooping cough, the last one, etc. etc. So we have naturally acquired immunity. But sometimes it's not natural. So we call it either passive or sometimes artificially acquired immunity. Artificially acquired immunity. Artificially acquired immunity. How many? 4. Artificially acquired immunity. So in artificially acquired immunity, we have two types. We have active immunity active immunity. So what will be active immunity? Active immunity is the one that involves vaccination. A weak form of a pathogen is introduced into the body of a person. And because that pathogen is weak, it is easily destroyed by the antibody. So the body produces antibodies, own antibodies against those pathogens. And then it is retained in the memory cells. So the body is exposed. Is exposed to a weak form of a pathogen. The effect is body produces its own antibodies against the pathogen. Remember, it's weak. Body produces antibodies produces its own antibodies against the pathogen. And now the structure of the pathogen is retained in memory cells. In memory cells. So the point weak should be emphasized. If you are going to be vaccinated against malaria, you are going to be injected with a pathogen, Plasmodium, but that Plasmodium should not be so strong so that it causes disease until you die. No, it should be weak so that the body can practice on it, can do some practice on it, kick it out, you produce some antibodies against it, and then clear it. That structure of the pathogen will be retained in the memory cells. Once it is retained, any subsequent attack again, the body will just go back to the memory cells and find out you are the same pathogen that you attacked us. This is the kind of antibodies that we produced. You cannot be able to defeat us. It's now going to clear the disease. So this is the principle on which vaccination works or immunization works. This is the principle of vaccination. In fact, this weakened form of a pathogen is usually called a vaccine. So when you talk of a vaccine, if you are told maybe at advanced level, if you start some medical courses, you are told to transport some vaccine. What will you be transporting? You'll be transporting pathogens, but they are in a weak form, so that they cannot cause a disease. The body practices on it, producing its own antibody, and then destroying it. So we call this one as active immunity, which is the major principle for vaccination or immunization. Then the last one is called passive immunity. From the word passive, pass. The body does not produce its own antibodies. It is obtained from a different source. We are going to find out the source. The body does not produce its own antibodies. So where does it get it from? But it is from a different source. So what are some of the sources that we can mention? For instance, serum. Remember, serum is blood without blood proteins, but rich in antibodies. So serum, for instance, you can develop serum this way. You can inject, let us continually use malaria. You can inject into maybe this rabbit. You inject a strong pathogen, Plasmodium, into the body of this rabbit. The rabbit is going to react to Plasmodium by producing antibodies. So antibodies will be produced. It's going to be produced in the blood system of the rabbit. Now, if you obtain serum from that rabbit, you obtain blood, and then now you separate to obtain serum. That serum will be containing antibodies against Plasmodium. So if there is somebody who requires antibodies against Plasmodium, you'll have picked the serum from the rabbit and then you just inject into that person or into that body, and is going to obtain healing. The other example is milk. The first milk, colostrum. Colostrum. This milk usually contains a lot of antibodies. So if a baby is fed on this milk, antibodies that are found in this colostrum will be passive serum. We have passed from the rabbit to the body of an individual. So again, from serum or through serum, through colostrum to the baby. Again, sometimes mother can also produce antibodies that will cross the placenta into the fetus before it is born. Antibodies across the placenta. Across the placenta. So these are the major categories of immunity. So in our next lesson, we are going to find out how, for example, if you get a question, how you can tackle it. But I hope you have understood. But again, if maybe you do not understand very well, we always have backup question. In our next lesson, we shall look at some of the questions before we embark on another lesson. Thank you.